Um, so this is about our protein docking methods called LZerD. Um, so I go through how the LZerD works. It is for pairwise protein-protein docking. And we also have a series of tools which also does multiple protein docking, disordered protein docking. I'm not sure which what kind of docking problem you have, but let me first go through the pairwise protein docking. Well, okay, as everyone knows that, okay, well, protein does interact with each other to do their functions. And then there are so many individual protein structures accumulated in PDB. And at the same time, you have experiments to confirm which proteins are interacting. So if you have a protein docking methods, then you could assemble those pieces together to make complex structure modeling. Yeah, so there's a protein-protein interaction network available. And recently, there are many works, emerging works for protein-protein assembly, well, design, artificial design of protein complexes. For that, computational modeling is really critical because we wanted to see how the complex would change by changing some part of the sequences. So, okay. So when people talk about docking, there are, they often talk about protein-protein docking, but the protein ligand docking, drug protein docking is another thing people consider as well, refer as by docking. So here I'm talking about protein-protein docking. In protein docking, it's, it's straightforward. Docking program takes two protein structures as input and then output bunch of assembly poses of two structures. So input is two structures, output is list of complex models, usually sorted by some scores. So here in this example, this is okay, interaction between subtilisin and eglin C. This is PDB ID is 1 CSE. That is on the left side. This is a crystal structure. And then, well, on the right side, I'm showing one example of a predicted model. Overall RMSD in this case is 2.74. It's not too bad, as you can see visually. Now protein docking, okay, will be, oh, okay. This is supposed to be the movie, but well, so here I wanted to show a bunch of models. So here, what movie was trying to show was the crystal structure is between this green and this gray structures, and the ligand protein now in magenta is docked to everywhere. Docking method usually goes with two-step procedure. The first step is to dock one of the protein, usually called ligand protein, to all of the different poses, all places with all different orientations. And this is like a conformation generation step. It will generate few thousands of or 100,000 of different conformations, depending on the interval of angles and shifting intervals you set. And then the second step is to rank them. It's similar in structure. It's also similar in protein structure modeling. In protein structure modeling, if you don't have a template for homology modeling, what the method usually does is that it generates lots of different structures, and then those are sorted by energy or some scoring function. Now, okay, so there are lots of methods actually. So protein docking has been studied from maybe around, okay, well, we can go a very long history here, but recent, well, the software started to be developed with from like 20, 30 years ago, and then our LZerD is one of them. We have this LZerD has some technical interesting aspects, but the overall logical flow, logical steps are same. Lizard will do the, well, build a lot of different conformations, and then in the second step, we have a scoring function to rank them. Now, LZerD, okay, LZerD is abbreviation of Local 3D Zelnicki Descriptor-Based Docking Program. We will, I'll explain a little bit about the 3D Zelnicki descriptor in the next few slides. But okay, so what we consider when we build the docking model is represented here. First, okay, so given the two protein structures here on the left side in purple, and then another protein in green. Well, we generate the surface representation of two structures and then spread the points, like representative points on the surface here shown as black here. Well, there will be, here I'm only showing a few points on the surface, but usually a protein have over hundreds of different points spread almost equally on the surface. Now, at each position at each points on the surface, we consider normal vector. Normal vector is a vector which points perpendicular to the local surface. So this tells you the direction of the surface, here represented by this arrow. Now, and then the 3D Zelnicki descriptor is a shape descriptor, mathematical shape descriptor. It has spherical harmonics in it. Well, I explain it later. So the Zelnicki descriptor captures the local surface, local landscape of the surface. Here, so the surface around the critical point is cut by a sphere of 6 angstrom, radius of 6 angstrom, and then those bumps in up and downs of this local surface is represented mathematically by the Zelnicki descriptors. So that is the second unique component of this LZerD algorithm.
Daisuke, can I ask a quick question?
Yes, of course, yes.
Is the 6 angstrom how far apart the points are spread across the protein, or is that a variable?
It's a 6 angstrom is a variable you can set when you before you run the program. But this is once you set distance 6 angstrom or whatever angstrom you want to try. This is 6 angstrom is unique. Well, it's same distance is used for all the points. So okay, normal vector and a local landscape by Zelnicki descriptors, and then we also consider interface area. So usually protein complex have large interface rather than touching each other with just a few residues pinpoint. And we also consider atom crashes, avoiding huge protrusions. Into another protein. So those 4 terms are considered in the initial stage of docking. And so what the scoring function is trying to do is to find the poses. Well, pose is scored based on how normal vectors are pointing each other with 180 degrees, close to 180 degrees, and then how well those local landscape fit each other. And also trying to also maximize the interface area. And also, okay, avoid if that pose have too many atom clashes, then those poses are rejected. So now I explain a little bit about this Zelnicki descriptor. So Zelnicki descriptor is mathematical representation of molecular, or actually 3D objects, and using a base function. It's based on the series function called called the Zelnicki Kantaraki spaces. So which has a form of in the first equation. Here, well, so what this Zelnicki descriptor can represent is the direction, the theta and phi, this is a spherical harmonics, which is also used for describing molecular orbitals, describing the way the electron distributes. And on top of that, it also has R, so radial function. So the function can represent is that starting from, well, relative to the origin of the points, which is in the case of protein structure, we put the origin at the center of the surface, local surface, and and then this function. Okay, so then this can represent where the surface locates in different directions, theta and phi, and the distance from that point. And so it has okay. So this is a series expansion. It has for each n, l, and m. Well, we have one basis, and then here in the second equation, this omega or which have fx and then zx here. Fx here is representing protein surface. So here fx is to describe a protein surface. We put protein structure to the 3D grid, and then mark the surface region by integer of 1, and then where the protein surface does not exist is recorded by 0. So 0 inside and 0 outside. So that is fx, and then that is well, kind of converted with Zernike descriptors, and then that has the one value of omega of index n, l, and m. Since, well, and we consider n up to up to certain number of n's, and that gives us like a vector of the basically the coefficient values of associated with this space function. So right.
Does your wave function incorporate the character of the surface, so the hydrophobicity or the charge?
That's a really good question. Actually, so for this, okay, in the application of protein docking, we only consider shape information. This is, okay, at this point, this is only shape matching. Actually, so the scoring function have two steps. First, we just pick up top 1,000 of structures by shape matching, and then later we consider that atom-atom interaction, more atom-based, and that can consider electrostatic interaction, hydrophobic interactions, and well, of course, van der Waals interactions, and in a more physical, and which makes more physical sense. So this shape descriptor is used for the initial screening in the first step.
Thank you.
So actually, Zernike descriptors is very useful for many things. It can be used for very quick PDB database search, or like ligand molecule search, compound search. Or here we are applying for protein-protein docking to identify the complementary surface shape, because this can be computed in rotation invariant way. In the last equation FNL, this is the rotation invariant descriptor of the molecular surface. Actually, if you take a norm over M, this descriptor can become a rotation invariant descriptor. So actually, we maintain a database called 3D Surfer, where we convert the old PDB entries in 3D Zernike descriptors at weekly basis, and then you can do the search, PDB search, against entire PDB actually within few seconds. It's not just looking up the pre-computed values at distance, but it is doing a real-time search.
So this, Daisuke, this would be just the outside of the protein. Would this would the shape also incorporate the active site? So if I wanted to search for a small molecule that would fit in my active site, could I do that as well?
Yeah, so actually, yes. Yeah, we can. Yeah, so for this, lizard is for protein-protein docking, but we also applied 3D ZenK descriptor for virtual drug screening. We have another software for that. So we applied it for pocket comparison, like active site comparison for structure-based function prediction, and also for virtual screening, drug virtual screening. In the virtual screening, input is pocket shape, or like the active sites you believe, and then we scan. You should also specify the database, drug database, like a zinc database or Kimball database, and then we can find the compound which matches to which have surface matches, and also physical chemical matches against the pocket. So yeah, this is really versatile methods for describing molecular surfaces, and then identify similar surfaces or complementary surface pairs.
Dr. Kihara, so I was wondering if there's a particular order that is more commonly used when doing such searches, because your figure shows like 5 orders.
Okay. So particular order of the searches. You mean order of the
No, sorry, I meant actually I used the incorrect term, but there's like 5 invariants, and there's like, I think there's more information in like as you go from B to F, there's more information in the shape that you get. So like I was wondering if there's a particular sort of benchmark where when you create these ZerDic descriptors for you to start as a search model, or like.
Okay, you mean the length of the vector.
Yeah.
So okay, so the base function of ZenK descriptors starts from just describing a surface of the sphere, and then as the n goes to this index goes further, then it starts to describe more in details. So and we try to find the best resolution. Actually, by changing n, I can control the resolution I consider. For molecular surface matches, you don't necessarily want to go too detailed, because in protein-protein docking or protein-ligand docking, atoms are you have a probably unbound form of the protein surface. So you want to consider some flexibility of the molecule, which can change surface a little bit. So you don't want to go too far to detail description, but of course you don't want to go too coarse-grained, low resolution surface matches. So for overall protein database search, we use n equal to 20. We tried several values, and then I find the value which performed best in the practical application. For protein docking, we used a little bit lower n because we also want to consider the conformational change upon docking, because bound form and unbound form of a protein differs a little bit, at least, or drastically. So we wanted to consider surface as like a kind of soft surface.
I see.
So here, right? So here, this is one example of how the surface region can be described by 3D ZenK descriptors. This is ribonuclease and Balstar complex, and then surface region is shown in blue and red here, and then the graph is showing the coefficient values of the 3D ZenK descriptors along the x-axis. This is the coefficient position of coefficient values. The first value is the coefficient value of the first term, and then second term, third term, and so on. this is okay. Bit difficult to see, but there's the white bars and black bars, and then those are the showing the coefficient values of blue surface and red surface. If you take the correlation coefficient value of this 3D Zernike descriptors, it's very high. It correlates very well, 0.96. So because they this is the interaction size, you expect that surface regions are more or less have a almost fitting and complementary shape. And we can, yeah, okay. So this part is represented. Now this is, we also did a benchmark for several other well, a dozen of protein complexes. On the left, okay, so the first PDB entry is a PDB entry of a complex structure, and then the in two PDB entries in parentheses is the individual structures solved so unbound form of individual structures, and the correlation value of the correlation value of Zernike descriptors at the interface in the bound form is on this column, 0.93, 0.96, all very high. If you consider the unbound interface, they still maintain a relatively high values. Well, this is well. This is showing that Zernike descriptors can identify the complementary surface regions. Now, for protein-protein docking, here what I computed is the shape complementarity of known bound interface between two structures. But that doesn't mean that. When I applied for protein docking, I don't know the interface regions. So for that, because of that, well, we segmented a protein surface by sphere. And then once a pose is generated, we computed the 3D Zernike based matches complementarity at individual points, matching points. Okay, so this is like an overview of how the LZerD docking method works. So we have an input of two proteins. One is called receptor, one is called ligand. This is a terminology in protein docking field. Usually, bigger one is called receptor, even if it is not, does not necessarily be a biological receptor. Receptor and ligands. Then we spread the points on surface, and then for each point on the surface, so we compute normal vectors and then 3D Zernike descriptors, and then do the well, conformational search. This is using some technique called geometric hashing. Geometric hashing is a fast way of doing the docking. And then at each docking pose, what it is computing is angle between normals. If two surfaces are fitting perfectly, it should have 180 degrees because it should point each other. And then Zernike descriptors correlation. This is checking the local surface matches. And then also compute surface area, larger is better, and the excluded volume. So this is a clash penalty. If penalty is so high, then we simply just reject them. And then we generate lots of models. Typically, it computes more than like 50,000 to 100,000 of models. And then we do the clustering to bigger ones. And then, okay, so actually, okay, so the ranking, yeah, so okay, actually, so there's two steps of ranking. One is shape-based ranking using this the Zernike descriptors and angles, and then the second step of ranking is using atom-based scoring function. Then finally, we have a sorted list, and then for top 10 or 20 or whatever you want, we do the MD molecular dynamics-based refinement to make sure that atoms are not crashing and in a reasonable physical orientations.
So, Daisuke, are you going to tell us about two things here? I'm interested in the atom-based scoring and how you put in biological things like we know there's mutants that interfere with binding, that kind of thing.
Okay, yeah. So
Okay.
I talk about the scoring here. So for scoring, we can also use molecular mechanics force field like a charm potential or amber potential, and we also tried that. But practically, that works fine. That works fine. But the score currently we are using is so-called knowledge-based statistical potential. And we use 3 of such scoring function. Those are called D-fire potential, GOP potential, and IT score potential. Those are developed by different labs. So what this scoring function is doing is it is basically counting statistics of atom interactions, angles, and distances of experimentally determined structures are actually the structures determined by X-ray. And if a model has agrees with the statistics, known statistics, this score will give you a better score. So this is so by considering basically okay, all this method is first for first counting the cases, counting the cases where 2 atoms in particular amino acids locate in certain distance bin and angle bin. So first it counts it, and then normalize it by a expected number of counts of such pose of position of 2 atoms, and then take minus log of it. So it is computing a potential from the statistics taken from PDB, representative structures from PDB. So these 3 methods performs in terms of scoring. It performs better than applying molecular mechanics force field, because these potentials are tuned for structure modeling purposes. So what we did was we ran those we ranked the models based on these 3 scoring functions separately first, and then aggregate the ranking from each of these 3 functions. Is computing sum of the ranks. So if a model, right, yes.
Sorry to interrupt, but do all 3 methods use the same training set or the same PDB basis set, or?
No, those are developed by different labs at different time. So they are using not exactly same data set, but their own data set. Well,
Is the data set updated?
No, so DeFi is published very long time ago. Even before, okay, I don't remember the date, but so they haven't updated the scoring function since their basically since the publication.
Okay, thanks.
So if a model is really good and then ranked first, ranked number 1 by each of these methods, and if you sum up that ranks, then that model have summed rank of 3. So 3 is the best rank. Then we sort that the models based on the sum of the ranks from this 3 scoring function, and then select top few models, what number of models you like. So this is okay. This is one example of how rank and actually the scoring function and accuracy correlates. So maybe we can look at this interface IRMST. IRMST is interface RMST. So this is RMST value computed only at the correct interface atoms. So x-axis, so here the first in the corner, this is the interface RMST on the y-axis. Oh, okay. Unit is angstrom, and then x-axis is the energy value by the scope function. So yeah, so and then okay, so this yellow and green dots, well, good models. This is a this is a good models or acceptable models in competition Capri competitions criteria. So as you can see, well, it's not, you know, by if you pick up the best GOP score model, then yeah, or if you take, let's say, top 10 best GOP score models, then actually you can take that includes, you know, best models within top 10. Same thing for DFIR. For this particular, each dot is the generated models for particular protein-protein interaction, particular protein complex. So there are, you know, few thousand of models evaluated here. In this particular distribution, model distribution, DFIR performed relatively well, because here, if you pick up, let's say, top 5 models by DFIR along the x-axis, then all of the models are acceptable. Same thing for IT score. Yeah, GOP didn't, okay, well, GOP is performing very well, but well, if you pick up top 10, well, majority of are acceptable, but some of these gray dots are included. So that's how GOP DFIR IT score performed in this particular protein complex models. LRMSD, this is same story. LRMSD is a ligand RMSD, so you fix receptor structures, and then you only measure how the ligand protein docks to the receptor protein. FNAT, this is a fraction of native contacts, so this is higher, it's better. So at the interface of protein model, it is counting the percentage fraction of atom pairs which are correctly recovered, native contacts which are correctly recovered by the model. So yeah, so this is, well, in good cases how GOP DFIR IT score performs. Okay, so far any question?
I had another question. So I didn't completely understand in this in the three Go, DFIR, and IT score. You said that you use the statistics from the PDB. How does that statistics sort of give you interface level information?
Okay, so actually, yeah. So this Go, DFIR, and IT score we are using designed for general protein structure prediction. This is not necessarily originally designed for selecting the correct interface of protein docking models, but it turned out, well, so although it's not, originally it's not for protein docking, we are using it because they seems to work well. So yeah, so GOP and DFIR is designed for, it's a more like a structure prediction. You have amino acid sequence, and then method will generate lots of different models, and then GOP and DFIR is supposed to select the native-like models, close to native models by checking angles and distance and the interactions.
I see.
Okay. So any well, other questions so far?
Actually, can you go back to where you put in the biological data? If, for example, you have mutants or someone suggested,
Okay,
in
okay, right, right.
or
Yeah,
whatever.
yeah, here, right. So, okay, so this can be done. So yeah, biological knowledge can be in the algorithm can be input in can be considered in two ways. One way is that we can, so for us one way is by considering post filtering. So we generate 50,000 lots of models without considering your biological knowledge or anything at all. And then once we generate lots of models, we only pick up the models that agree with the known interface residues or distances or other knowledge you have. Another way is to restrict the conformation search by considering the interface. So if you know that residue, couple of residues at the interface, we don't need to explore the other positions on the surface, and we only do the build the docking models, just focusing on that regions. So here, yeah, in this diagram, it is, well, this is trying to explain the latter way. In our server, maybe Charles can help me. So when we input we also established the lizard web server, and in lizard server, you can specify residues which are at the interface or residues like a distance of residue pairs from receptor and the ligand. And I think those information is used as a post filtering in the website.
Yes, correct.
Yeah. So, okay. If there's no other questions, I can show you well, two ways to use our methods. One is the web server. We recently developed it. So here, okay, here, this is a web server. So what you can do, you can just simply input two PDB structures, and then there's a panel here. Okay, maybe it's a bit difficult to see, but you can also specify which residues are supposed to be at the interface, and or if you know interacting residue pairs between two proteins, you can also specify those two residues and also the distance between them. If you have, if they're not interacting but known, well, like cross-linked or something, then that information can be also input here. Then, yeah, then it will give you, well, so you will receive email back in a few hours, or I don't know, a couple, well, it can be really quick, but if proteins are very large, then you will receive it in a few hours or could be a day or so. Then the models are sorted by the scoring function that the ranks, the sum of the rank scoring functions. And if you click each of them, then you can visualize the model. So in the first panel, what second panel is showing is if you place a receptor protein in the middle, here shown in cyan, where the center of the ligand protein locates. So you can see among the, I think 100, okay, among top 50 models, you can see where the center of ligand protein locates to see how they distribute. And then you can download well, you can download all the protein PDB entries. Yeah, that's a web server. Or you can also download the program on your computer and run them locally. So that is a pairwise modeling using LZerD. Okay, so that's
the end of your presentation.
yeah. So yeah, that's about lizard. I just show you some other methods, multi-lizard and so on, just briefly. Okay, show yeah. Well, show you in case you are you have application for that. So this is okay. This is our recent work on deep learning based model selection. So previously we used GOP, DFire, and IT score for ranking models, but we can also, this method uses deep learning to capture known observed like distribution of atom position at the interface. So what it does is that if you, well, you input proteins complex model, and then this method will capture atom distribution at the interface and tell you if a model is likely to be correct or incorrect. This is okay, a very recent work. This is the graph is showing performance of GOP, IT score, and relative to this new methods DOLE, deep learning based scoring function. For pairwise interaction, DOLE performs better than individual methods. So for pairwise models, we consider DOLE also as an additional component of the sum of the ranks.
Okay, sorry, Daisuke.
No. Yes.
So I can't, it's too small for me to read. What's on the X and Y axis here?
Oh, okay, okay,
Sorry.
right, okay, okay.
I
So
can't see it.
sure. Yeah, x-axis is the okay top ranks considered. So what we have is we have a decoy model set of I don't remember the number of target complexes we used, but for each of the target protein target complex we have hundreds of models, and then those models for a particular protein complex is sorted by each this scoring function like GOP, and then check if top n ranks, like let's say up to top 5, include any good model or not. That is so and hit rate is showing that how many target we a scoring function like GOP could rank correct models, acceptable models within top 5 or top X ranks.
So if it was perfect, it would be 1.
1, yes.
Right, okay.
So, okay, this is, okay, so this is I'll just show you a couple of extension of pairwise lizard. This is multiple lizard. So if you have 3 chains or 4 chains up to about 6 chains, you want to dock, assemble, then multi-lizard can do that. Yeah, so here, what it does, well, this is the workflow, but well, just briefly, what it does is first it docks every pair of proteins, A, B chain, A against B, B against C, C against D, and so on. And then in the second step, it will assemble pairwise interaction, pairwise models together to build a full chain model. Yeah, so this is and then do some refinement. So this is for multiple chains. Okay, well, so this is some examples. So for example, for this 2-AZE, this is a 3-chain complex. I don't remember the name of the protein, but this is a PDB code. And the 3-chain complex is assembled by from pairwise interactions, pairwise models of AB and then also AC, separately originally modeled. And what it's showing, this rank is showing is that the rank of pairs selected to assemble the 3-chain complex. So So for AB pair, the best scoring model is selected. For AC pair, the 918th model is selected, and then that pairs assembled pair have a RMSD, very good RMSD of 0.73 against the crystal structures. So now actually, by looking at the ranks of a pairwise interactions selected, we can also predict assemble order of protein complex, because what it is showing, the rank is indicating that how well A and B of this particular model fit each other relative to other models, other possible AB interaction models. So by looking at these ranks, we can also consider predict which one docks first. This is published in a different paper. Okay, later. Well, this is some examples of multiple protein complexes. Well, I can probably skip here. So here, this is about docking order. For example, this urease accessory complex have, okay, so 6 chains in a complex. And then it docks in this order. Chain F and H docks first, and G and G prime docks, this is the homodimer, and F prime and H prime docks separately. And then finally, okay, then at the second step, well, F and H will form a 4-chain complex. And then finally, G and G prime will dock at the last step. So this,
is that a prediction?
No,
or is that...
this is a known. In this case, this is the experimentally identified path, assembled path.
Okay. Mm-hmm.
And that can be predicted also by checking the ranks of pairs used to dock this 6-chain complex.
I have one other question there. What if, I mean, there's an example here. Some of the binding surfaces cross various complexes. You know, you don't have the binding surface for the third protein until the first two come together. Can you, is that what makes it 918th in the rank or something? Give us a space or what?
Yeah, so yeah, I think so. I don't remember individual cases, but if ranks is very low, like this case, 5077, it's really low. Probably this model pairwise model do not have very large interface, and that's why the score was very low.
I just wonder if you knew that worked or not. Who knows?
Yeah, so actually for a larger complex of 6-chain and 7 chains, this algorithm started to have some difficulty because making pairwise models is getting difficult for cases with very small interface between each other. For 3 chains, for 3 chains and 4 chains, this method works very well. But when it comes to 6 chains, 7 chains, 8 chains, some of the chains are not even touching each other, but the algorithm doesn't know unless you tell the algorithm that, okay, chain A and D never are far apart on the complex. Algorithms still try to dock them, and then consider that possibility too. So that makes it a method bit difficult actually. We are trying to improve these methods right now, but that's one of the difficulty we face. This algorithm faces. Okay, well, yeah, okay. This was there was a movie also. This is showing how the pieces assembled together in a docking pathways that could be predicted by our methods, but since a movie seems not working, I'll skip this. So other extension we have is for docking disordered protein. If you have a disordered protein interacting with ordered protein, we can do that. So that was questions a common question I usually had when I present our protein docking methods, the question is, how do you deal with disordered protein for docking? Previously, I needed to say, no, I cannot do that, but now we have methods to do that. So for docking disordered protein, the difficulty from algorithm point of view is that the disordered protein does not have a structure. So I don't have a structure to dock. But this is done by Lena Peterson, graduated about 2-3 years ago. So the algorithm of this intrinsic disordered region lizard, IDR-LZerD, is what it does is, okay, so for disordered protein, I assume I only have a sequence information because it's disordered. For receptor protein, I need to have a structure. Now for disordered protein, I predict actually, I cut the sequence into pieces of, in this case, 9 residue long pieces, and then predict fragment structure of 9 residue fragment overlapped with 3 residues, and then dock them separately, and then connect those. So here, there's the blue, yellow, red regions predicted, whose structures are predicted separately, and then I dock each of them individually, and then finally, on the protein surface, receptor protein surface, I try to connect them. In that way, we can also dock disordered protein. Like in this case, this is our most extreme cases. I think, okay, so here, green is a native structure, crystal structure. Red or orange is our model. You see that this turns, but but visually you can see that we can dock the 69 residue disordered protein on the receptor surface. Fairly well. So we can also do so using this method, we can also do the peptide docking. If you have peptides, we can also dock them. For peptides, we only need a sequence. Protein docking can be also applied for fitting individual structures to EM map. So here, here, suppose I have a EM map, and then individual here, individual structure needs to be known or modeled. Then we assemble those pieces together into the low resolution map. So this is just some examples, and then basically that's it. So we have pairwise docking. So I didn't shown today, but we can also predict the docking interface regions from more bioinformatics sequence analysis. And then we can guide the pairwise docking by predicted interface regions. Okay, so there's, right. So this is binding site prediction methods. And then we extended pairwise to multiple and EM fitting and also disordered protein docking. So depending on what kind of protein you have and what kind of information you have, we have a several set of tools for hopefully helping or at least providing a hypothesis of protein-protein interactions. Okay, so all right. Okay, maybe I talked too much.
We interrupted a lot. So questions from the group.
Dr. Kihara, well, first of all, thank you for doing this. I'm interested in, actually experimentally for my own work, I'm interested in that BindML and PI-LZerD sequence. I don't know anything about BindML. I know with my 2 proteins, there have been some computationally determined potential residues. Okay. I'm guessing though it might be better to just use BindML rather than try to feed those in, since I didn't do that work, or? Okay,
so BindML, sure, we can. Okay, BindML has a server, and I hope it still works. But BindML is performs well. I cannot say that it performs much better than other existing methods. But it is using kind of original idea of checking like mutation patterns from multiple sequence alignment at the protein interface. So it can probably give you like a orthogonal information with some information you already have. And with that information, we can run LZerD on the web server, and then guide the docking with the predictions. PI-LZerD, yeah, this is another method we can apply, but currently this needs more computational power to do it. So practically, maybe we can use the LZerD web server.
Okay, so could you take, so like say you had binding residues that you don't really know, maybe they're more putative, you don't necessarily know like the distances between residues that may interact, and then do more of a permissive search, or I guess that's kind of vague.
Yeah, I think so, yeah.
Okay, okay. The other question I had was, and this is just a really basic question, I don't know anything about geometric hashing. I was curious if you could say any more about that.
Okay. Yeah, geometric hashing actually first record different poses in a database. So actually I may have, so in geometric hash, so to define a pose, you can define a pose of a 3D object by specifying 3 points on the position of 3 points on the surface. Or if you can also specify orientation of a molecule by specifying 2 points on the surface with the normal vectors. If you tell 2 points and normal vector should point some certain direction, then you can fix the pose of one molecule. So what geometric hashing is doing is, so in our case, since we use normal vectors, for every pair of points on the receptor protein, we record the pose of the receptor protein, and also surrounding points on the surface information to the database. Now that is the first step of the geometric hashing. So all the orientation of receptor protein is already hashed, or kept in a database. Now I have a ligand protein, and then I do the basically same thing for every pair of points which are close enough, or not too distant, or not too close to each other. I pick up the two points and then compute, consider the orientations, and then check what kind of position of surrounding surface points in that particular position. And then I try to find if such distribution of the surface points has ever recorded in a database or not. And then the hashing gives a very quick search of that particular orientation. And then we pick up that orientation from a database, and then if that matches very well, sufficiently, then we build a model, 3D model, and then make sure that there is no atom crashes and so on.
Okay, thank you.
Other questions?
I was interested in your comment about trying to use your this paradigm to explore an active site with a library of ligands. What would I go look up to learn more about that?
Okay, sure. So let me. So we have we have a web server, or actually so this is called PatchSurfer. what it does, so it was published in JCIM in 2016. Yeah, so yeah, please take a look at this paper.
Okay.
But what this paper does is basically the idea is very similar. So here, suppose you have a known binding site in a protein, and then then I spread the points on the surface of the receptor, and then do the same thing basically. Represent and then cut the surface by a sphere, and then compute 3D Zernike descriptors that gives us the local surface information. And well, we also consider because this is a ligand protein interaction, we consider hydrophobicity on the surface, electrostatic potential on the surface. Actually,
Okay.
so in the protein docking, I said that we only consider shape information by 3D Zernike descriptors, but 3D Zernike descriptors can also consider the other values on the surface too. We can just map the values, hydrophobicity values or electrostatic potential values on the surface, and then compute 3D Zernike descriptors for that distribution. We can do that too. So in this PL protein ligand patch surfer, we consider the hydrophobicity, electrostatic potential, and in the newer version, we also consider hydrogen bond donor and acceptor positions. And then those are all converted in 3D Zernike descriptors, and then do the same thing for each of the ligands in the database. It takes some time to compute it, but we already have that computed files of Campbell and zinc data set.
Okay.
Then,
yeah,
and then do the basic matching. Then identify the pairs of surface between ligand and then patch in the protein binding site, which agrees, matches very well, and then combine the score.
And then okay, well, very good. Thank you. I will look that up.
Yeah.
I have a question.
Yeah.
I have a couple of questions left. One on that is, what if you have your own library that you want to put in? Is that possible to do?
Yes. So I think probably this package has a tool to do it, but yes, it's possible. Of course, it's possible.
Okay. Other questions before I ask mine?
Sorry, I thought of one more. Can I take, so for like basic lizard, can I take, and I'm assuming the answer is yes, but I don't know, two proteins, a receptor and a ligand, and say the receptor protein has like a small molecule ligand bound in the active site, and I have a PDB file of that. Can I then use the tools available to look at how the ligand protein binds, or how the small molecule might affect binding?
Okay. So yeah, that's a good question. Actually, in terms of shape, this method, the lizard can consider effect of how ligands affect to the binding. But the scoring function we use, the GOP, D-Fire, and IT score, they don't have a parameters for actually non-protein atoms. So in this scoring step, in the last scoring step, effect of in the heteroatoms are not considered. So to be able to consider ligands, additional heteroatoms, we need to run molecular mechanics based force field or something else, rather than using the protocol we have.
Okay.
So it's not possible to do it, if you just download the package, or if you submit the models to the website. The shape is considered, only shape is considered, but not the chemical, physical chemical interactions.
So that they must apply for post-translational modifications like phosphorylation or ubiquitization or any of that.
Right. That's difficult. Yeah, that's difficult. That's okay, only currently only shape is considered, and we need to think about individual. I mean, so we can run MD or something to properly consider modifications, but not just running this package as it is.
Okay.
Yeah, that's actually a weakness of this method, actually.
Okay, thank you.
Other questions? Does Charles want to add anything?
Only if someone has questions related to the large-scale flexibility of protein-protein docking.
Yeah, so Charles is currently, so this method is LZerD is using a soft representation of protein surface, but that is really just change of like side chain orientation or some small changes in backbone. Okay, in terms of RMSD, LZerD soft surface representation can only consider up to like a 2 angstrom, probably maybe up to 3 angstrom RMSD difference. But if you have a huge conformational change in either one of the protein, any of the methods currently available can handle those. But Charles is now trying to consider a huge conformational change like calmodulin. You know, calmodulin have a dumbbell shape, but when it talks to receptor, intermediate alpha, internal alpha, long alpha helix will be bent, and then kind of wrap around against the receptor protein, and those kind of large motion conventionally not considered, but now Charles is developing those kind of methods. So if you want to dock a protein with huge flexibility, we could consider. Yeah, let us know.
That's possible, yeah. Okay. And so that'll be done any moment, right, Charles?
Yeah, it's all like a method itself is already there. He's just trying on benchmarking on various different proteins now.
Okay.
Unfortunately, it is very computationally intensive.
Yes, I'm sure. Any other questions for our guests? Or can we come ask you questions when we realize we didn't really understand, even though you presented it, we thought we understood?
Okay.
We'll,
yeah, sounds
we'll
good.
call you up by Zoom and say, actually, we don't understand Zernicase. No, descriptors, but Otto does.
Okay.
Right?
Well, I find myself having to brush up on a lot of things. Yeah.
Dr. Kihara, sorry. So you had mentioned that this this presentation with your voiceover will be up on your website at some point. I'd be interested when it does go up, just to review it.
Sure. So yeah, so we have we were interested in starting a YouTube channel. Actually, one of the students that suggested it, and I thought, oh, that's a great idea, but we don't have the contents. So I thought this could be a well, if it looks okay, then yeah, we are interested in uploading it as a first content.
Okay, yeah, that'd be great.
Jill certainly is familiar with YouTube at this point.
Yes.
Oh, okay.
All right, I guess that's it for the presentation, unless anyone has any last minute questions. If not, thank you very much. We really appreciated the taking.
Yeah, thank you very much. Yeah, thank you very much.
Thanks, Dr. Kihara.
Thank you very much.
Thank you.
Mhm.